But I want to welcome you all for the first Wildlife Wednesday with Center for Wildlife Studies. I'm Jack Hopkins. I'm the president and associate professor with Center for Wildlife Studies, also co-founder. And today I'm pleased to introduce Dr. Jeff Stetz. He's the director of research for Center for Wildlife Studies. He's just came on board a few months ago. We're really excited to have him. For the past 20 years, Jeff has worked primarily on large-scale studies of abundance, population growth rates, and resource selection for large carnivores in the American Intermountain West, Canada, and Alaska. These projects have included his master's work developing non-invasive monitoring methods for grizzly bears in Montana, and his PhD work explaining and monitoring population performance in grizzly and American black bears, both degrees he got at the University of Montana in Missoula. In addition to providing the first population-wide abundance estimates for grizzly bears in northern Montana and the first density estimate for American black bears in and around Glacier National Park, he's also worked on developing new population monitoring methods for mountain lions and river otters in North America and 2 bear species in the Russian Far East, as well as conducting rigorous evaluations of numerous monitoring methods for black bears across northeastern North America. For the past 5 years, Jeff's been in Alaska. He's worked as the wildlife research coordinator for an area the size of Montana, spanning from the Yukon border across the Aleutian Archipelago. There, Jeff worked with a team of biologists, collaborators, and graduate students to address wide-ranging conservation and management issues. These include population dynamics of genetic population structure of caribou, migrating moose vehicle collisions, mitigating moose vehicle collisions, developing epigenetic or molecular clocks for bears and caribou, exploring new monitoring methods for wolves, and the ongoing project that he'll discuss today, which uses contemporary non-invasive genetic sampling methods in conjunction with spatially explicit capture-mark-recapture models to better understand population status and performance of grizzly bears in central Alaska. While working for the state, Jeff taught courses on capture-mark-recapture for CWS. He has 2 coming up here in September and then another in October, where he strives to bring real-world experiences to help students and professionals around the world like you improve your understanding of the important field methods and statistical tools used to estimate density and other important population parameters used to inform wildlife conservation and management. In addition to teaching, Jeff's responsible for both our research and science support programs and is an associate editor of the Journal of Wildlife Management, a certified wildlife biologist, and a scuba diver who enjoys a good hearty IPA and the world's best cat, which he'll probably reference several times throughout his talk. Welcome, Jeff.
Thanks, Jack. That last note was meant for you, by the
way. a few quick ground rules just to add to the few that I said in the beginning, and I'll say those again. Let's keep our audio off, and if you have questions, please jot them down or do whatever you need to do to remember. We'll ask them at the end. We'll try to field as many as we can. Those questions can be related to his talk. They can be related to his past work, the ongoing work that he's going to be talking about today, obviously, but also the courses that he offers. One in capture-mark-recapture. It's the intro level course, and the second course in the series, a little bit more of an advanced course that he uses a lot more of the spatially explicit capture-mark-recapture methods in. So please keep your questions to the end, and then we can put them in the chat, and I'll go through them and I'll ask them, or we can, depending on how many questions we have, I can have you ask them. Let's just see how that goes. We'll sort of play it by ear. With that said, Jeff, why don't you take it away?
Thanks, Jack. And just because I am in Alaska and sometimes our bandwidth is not what it should be, I may actually turn my camera off just to help speed things up a little bit. And can you see my screen, Jack?
Yes.
Okay. And which screen are you seeing?
Now I am seeing I'm seeing your notes screen. This is like your notes and your, there we go.
Let's try
You got to change that setting.
that one. How's that?
Let's see. Excellent.
Okay. All right. Thanks again, everyone, for tuning in, and Jack for the generous introduction. As the title suggests, we'll be talking about grizzly bear population dynamics in Central Alaska using DNA-based capture-mark-recapture techniques. And then we'll move into a little bit more discussion about the coursework that CWS offers towards the end and how these two are related. And hopefully this thing behaves today. First and foremost, this is the work I'm going to be talking about today was and continues to be a very collaborative effort. Some of these partners are like National Park Service, Alaska State Parks, BLM, just gave us permits to do work on their lands. As you'll see in a few minutes, the study area was a pretty substantial chunk of country, about 60,000 square kilometers. So we definitely overlapped with very broad-ranging management and habitat uses or habitat types. The primary collaborators for this project were the Alaska Cooperative Fish and Wildlife Research Unit at the University of Alaska Fairbanks, AITRC up here in the upper right, that's Ahtna Intertribal Resource Commission. That's a nonprofit, tribally affiliated group, conservation group in Glen Allen, Alaska, primarily. And then the Alaska Department of Fish and Game, ADF&G, where, as Jack mentioned, I worked for the previous 5.5 years or so as the research coordinator for Region 4. And at the bottom right, Wildlife Genetics International continues to be just a fantastic resource for folks interested, like Jack and myself, long-term projects using non-invasive genetic sampling in particular to address all sorts of questions related to wildlife population, demography, connectivity, and so on. So a bit of a background on the project that we're going to be spending most of the talk on. By about 2018, which is when I came up to Alaska this round, biologists at Ahtna Inc., and I'll explain a bit more about some of these entities as I go, started collecting bear hair samples for genetic analysis on Ahtna lands. Ahtna, there's a very different model for tribes or native entities in Alaska than most people may be familiar with in the lower 48 states. In Alaska, they're native corporations. They're actually established as for-profit. These are developed much later in the history of our country. These came about in the early 80s. It's a very complex issue. I'm not even going to attempt to explain it to folks. But traditionally, Ahtna lands were about the size of Ohio, coincidentally. We've got at least one person on the line, or two people, I guess, including Jack from Ohio. So a pretty substantial area. Currently, there are about 2,000 shareholders. Like I said, this is considered a for-profit corporation, and with diverse interests, whether it's developing forest products, construction, so on. And collectively, they own about 1.6 million acres. Although, unfortunately, I think for everybody, it's kind of a patchwork design. It actually covers a much, much larger area, nearly the entire size of the Ahtna lands there, highlighted on the map in the lower right. And for the shareholders, they came to the board and were requesting more information about the local bear population. It wasn't terribly specific, as often questions begin. Just more information was the request, and that evolved eventually into the project that I'll be talking about. At about this same time, ADF&G, so Alaska Department of Fish and Game, was initiating their own bear study. Actually, the third iteration of a demographic study, and as you can see here in the upper right, the heavy black line represents Game Management Unit 13, which that's 60,000 or so thousand square kilometer area. You can see Anchorage there in the lower left. This is a very high demand region due to its proximity to the population centers in Alaska. The whole entire state of Alaska, which, as many folks may know, is about a third the size of the continental United States, has less than 800,000 people, but about half of them live in Anchorage. So in terms of caribou and moose harvest in particular, GMU 13 is nicknamed the breadbasket of Alaska. So it's a very, very important area for harvest, particularly of those two prized game species, caribou and moose. So going back at least 20 some years, the state has been directed by the Board of Game to reduce density of brown bears or grizzly bears, same thing, in this region to help maintain high levels of ungulate harvest. And my issues are not with management. Our job is to provide the best available data. So these studies were done early on in the early 90s initially, and then a second round was finished up in about 2019 or 2020. where we looked at this small, you can see the white checkerboard. It's a small area, about 3,000 square kilometers, where pretty intensive surveys went on using live capture-based methods. So we flew around and darted bears from helicopters, although the fun stuff we think we do when we're actually sitting in the office. And there were two primary objectives. One was to use a capture-mark-recite approach, where it's aerial-based. You go out and survey those little white blocks about 6 times over the course of a week, looking for the bears that we'd collared over the previous 3 or 4 years. And then, much like the models I'll be talking, you can estimate the overall density for that area. And also using the vital rate information that we collected from those collared bears over that 4-year or so period, looking at reproductive rates and survival rates. And as in the previous study, these can be used in a projection model where you estimate or predict what population growth rate will be for this population, assuming things stay more or less the way they are. This is different than a realized population growth rate, which you could obtain from the mark-recite, the density estimates. Look at how things actually did change over the interval between those surveys. So very similar objectives in a sense, 2 different ways of getting there, and ideally they will more or less align. And around 2020, biologists from most of the larger partner groups I mentioned before, so Adna Inc., Fish and Game, ATRIC, as we call it, and the Cooperative Research Unit at UAF, decided to develop a parallel study to the ongoing one from Fish and Game. In this case, our overarching objective was to use non-invasive genetic sampling, which I'll describe more in a moment if folks aren't familiar with it, using these genetic methods to complement and provide additional inferences on the ongoing Fish and Game study. As many folks probably recognize, the opportunity to have ongoing concurrent studies like this is pretty rare in our field, I would argue. Usually we do a snapshot abundance or density estimate or look at population growth rates, but it's pretty rare to have the same population, same time, same objectives with entirely independent data streams. Now, these can be combined in some ways, but they still are independent. And so the opportunity to compare inferences from these different methods, which I'll go into a bit more shortly, was just too good of an opportunity to miss, especially given some of the experience that some of us brought in terms of this genetic sampling. Our specific specific objectives for the study were, again, to estimate density and abundance, which not the same thing exactly. We'll talk more about that in a minute as well. And then potentially to model the variation in density across this pretty large, diverse area. Another issue I would argue with having such a small study area as the one there in white is it's difficult to capture. It's a very heterogeneous landscape. Everything from expansive areas of permanent ice and snowfields and glaciers where there's really nothing living, some large water bodies, on up to some pretty high quality bear habitat, whether it's related to spawning salmonid water bodies or just very dense ungulate populations in some places. So to think that you could extrapolate from a small study like this 3,000 square kilometers to a much larger area leaves a lot to be desired, arguably. And the other thing, like I mentioned, it's a rare opportunity to contrast estimates and inferences from a pretty broad suite of methods, including the mark-recite methods I mentioned and the projection models. We also had a graduate student working on stable isotope, looking at diet of bears in this region. We can do resource selection function modeling, or one of the variants of that using our GPS collar data. So how well do these inferences align? If we only had 1 or 2 of them, would we think the same things are happening in that population? And then hopefully to use the detection data from the genetic sampling to design a longer term study or monitoring program, the faster, better, cheaper approach that we believe the genetic sampling can provide to these more snapshot intensive. The collaring study was roughly $1.3 million and took 4 years to complete. Whereas in a single season, you can do these genetic sampling studies and obtain potentially even more reliable and more robust estimates of some of these demographic parameters that managers really care about. So how do we do this? How do we go about addressing these objectives? So typically, I would argue that studies should always begin in the same way. What's your question? Not, I want to go out and hang collars on bears and then figure out what to do, or put collars on caribou and figure out what to do. But what is your question? And then we work together and develop specific and the appropriate sampling analytical methods to address those questions. In our case, things were a little different because our objectives included exploring these specific methods. So we were off to a bit of a head start in that regard, and did have a pretty big foundation to build on. So getting into market capture itself, which is really the foundation of this study, these methods revolve around the reality that we observe animals imperfectly. To put another way, that the detection probability of any given individual in a population in a given time and place is less than 1. So generally, you can consider CMR models as a suite of methods that may be appropriate, if designed properly, for estimating these demographic parameters, despite this imperfect detection of focal species. By demographic parameters, this is just a partial collection of the things that we can estimate from the kind of sampling that I'll go into more here in a second, whether it's abundance, density, population growth rate, survival, recruitment, transition rates, and other things that, again, are very valuable to wildlife managers. CMR, or these estimates, statistical estimates, are different than a census. A census assumes all animals or individuals are detected perfectly. So that'd be a detection. P is the shorthand for detection probability. So if you had perfect detection, that would be a census. If you look, you find every time. Unfortunately, things aren't quite that simple as a rule with wildlife, or humans for that matter. In CMR estimates, or any statistical estimate, is also different than an index. Things like track counts or scat counts that may or may not reflect measures of uncertainty because they're not a statistical sample. They're just indices, which can be valuable at times, or at least help develop questions about what's happening with the population. But typically, they're not reliable enough or powerful enough to really give us insights on what's happening in the population. So, of course, all these models have assumptions. All models do. And one of these being, for example, that the marks on these animals are permanent, and meaning they don't change over time, and that we always read them correctly. And you can imagine where the challenges would be if marks changed over time or were lost, how that could affect or bias your sample. That said, many models, both the ones I'll discuss today and others, have evolved, I would say, to provide some ways to relax these assumptions, whether it's using covariates, say, to measure variation in sampling effort, looking at males and females differently, ages differently, that kind of thing, that we can now model those directly and get away from some of the assumptions that were inherent in earlier models. So CMR, capture-mark-recapture, used to be literal terms, where you capture a sample of the population of interest. Here, that's known as a capital M. And we mark animals. Sorry, my PowerPoint's taking off without me. So we mark animals, which can be either individuals or groups, depending on the specific model you're using. It may be possible just to have, a generic mark on the animals we handle, release those back in the population, and then at a second time, or potentially multiple times, we recapture and conduct second or tertiary, whatever, samples of that population. So the very origins of these models are incredibly simple. And if memory serves, this goes back to, I believe it was 1786, when Laplace tried to estimate the abundance of humans in France. And then these were refined in the early 1900s by two guys named Lincoln and Peterson, hence the name of the model being Lincoln-Peterson estimator. So again, M being the number. So if you go out and conduct a sample of our little fake population here of beavers, we captured 4 and released the first time, went out again, caught 5 more. 2 of those we caught and had marked previously. And so those are the numbers we need, believe it or not. What this provides is a very simple equation where, using our little fake example, I wish things were this simple in real life, we'd estimate the population to be 10 overall. And then this, of course, can be extended to provide or to allow additional samples and to calculate uncertainty of those estimates. So we would say 10 plus or minus whatever they may be, 2 animals. So in reality, especially with these earlier models, what we're really estimating is the detection probability, or probability of detection. We don't directly estimate the number of animals in the population. It's basically a ratio estimator. So we derive that estimate of detection probability, that p hat we see. We know the number we detected, n, and you essentially correct for that by dividing by the detection probability. which was pretty obvious in that previous example. So in our case, how did we mark bears in our population? That's obviously a very important part of this process, not only because we have to know who we're talking about, but knowing that the marks follow those assumptions of the model, that they're always read correctly and are never lost. In our case, we deployed what are commonly called hair traps. I imagine a lot of folks are familiar with this. So it's been around since the mid-90s. It's a method that takes advantage of the incredible olfactory acuity of bears, which I've estimated off the back of an envelope at around 2,600 times as powerful or as sensitive as humans. So we take advantage of that and their drive to find food. So we use these genotypes as marks. In theory, these would never be lost. An animal doesn't change its genotype. And given how quality or how reliable the lab methods have become, we can have a lot of confidence that these genotypes are reliable across time as we resample the same individuals. So like I said, these are key assumptions of almost any market capture model, whether it's a spatial model, open, closed population, whether you're looking at abundance, density, doesn't really matter. Just a bit more about the methods themselves. Like the previous photo was a single strand of barbed wire strung around a bunch of trees. We have looked at other methods. This was initially developed, as far as I know, in Yukon. Potentially BC used it before that, and then we modified it. We got to work with some captive bears here in Alaska, and just found new ways to be able to sample in these more or less treeless environments, of which there's quite a bit in Alaska. Another important thing is the overall goal is really to either minimize the variation in detection rates of these individuals and also over time, and to do things like minimizing behavioral responses. So folks are probably familiar with the ideas of being like trap happy or trap shy, for example. And to try and reduce those effects, we used a non-rewarding lure or an attractant, basically just rotten cattle blood, that we know can bring in bears from pretty good distance. Now, the fact that it's non-rewarding means that you're not biasing the behavior of this animal going forward. If it gets a food reward, it could eventually develop a trap happy response. So you're more likely to recatch an animal than to catch one the first time. And so there's an obvious bias there. Conversely, we didn't want to have a trap shy response or a trap aversion response. So we modified the attractiveness of these sites by adding secondary scents that we changed, and we moved the sites between our sampling occasions, just to try and keep the bears guessing. There's no harm to them in visiting these other than some wasted time. They might enjoy it, in fact, just checking out their backyard, so to speak. But there are concerns with declining efficiency of these sites. So we did what we could to reduce that without inducing a lot of variability across time or space, for that matter. But this doesn't answer the question of how hard did we have to look? How do we determine what the sampling effort had to be for this study? So this is, of course, a function of a few things. One being the detection rate of individuals themselves, and another being the desired precision of our estimates. Precision is not a one-size-fits-all issue. This idea that a coefficient of variation of 20% is good for every objective is just it's terribly misleading. So we talk to our managers and try to decide what a reliable or a useful target would be in terms of precision for this particular population. And that's also, of course, a function of the models being used. Fortunately, there are some very powerful tools that have been developed in recent years to help develop study plans, essentially, design our studies. So moving into the specifics of the models that we were using just a little bit, it's pretty obvious that detection is inherently a spatial process. That concept I mentioned before of trying to minimize variation in detection. Traditional models up until the last 20 years or so simply looked at whether or not you detect an animal in a given time. There was no role of space in there. But as you can see in this diagram to the right, if you picture each of those bear images as being the activity center or home range center of an animal, you can see how variable their exposure is to those little red pluses, which indicate or represent a sampling site, a hair trap in our case. So if you're a bear living in the center of your grid, of the sampling grid, you likely have a much higher chance of being detected than if your home range barely overlaps the sampled area. But without a spatial model, that information is not available in the data that you feed the model. So these seeker models, spatially explicit capture-recapture, I like to abbreviate that as seeker. It's quite a mouthful otherwise. These models, what they actually are estimating is, unlike traditional models, where I said they're estimating that nuisance parameter of detection probability, seeker models directly estimate the density of animal activity centers in a definable area. So this approach directly incorporates information on where you look and where you do and do not detect individual animals. And then you track those detections over time. Another advantage of this would be the temporal nature, that with traditional models like the Lincoln-Peterson I showed earlier, you could detect an animal at 4 different sites in one sampling occasion, and that still counts as a 1. Just a single binary, yes, we did detect them. These seeker methods take advantage of all the information contained in those encounter histories, as we call them, of where and when we detect individuals. So another way to look at this, sorry, PowerPoint. Another way to look at this, how seeker views a relationship, or relies on this relationship between detection probability and the distance between each animal's activity center, which of course we don't know, but is estimated by the model, and these individual detectors, in our case, again, hair traps. So as you can see there in the lower right, if the activity center is denoted as that black dot in the center, the animal is expected to use the areas surrounding the activity, the home range center, more often than more peripheral areas. And so there's this expected relationship between detection probability and that distance between sites and home ranges. So the methods have been developed that allow us to use existing information. In our case, we were able to use data from those GPS collared bears that we had on the air just preceding our study, and then use published estimates of things like detection probabilities to run a series of simulations to help us hone in our design, so to speak. So in our case, we only looked at female grizzlies because in case of most mammals, females have smaller home ranges than males. And so if you're sampling with enough intensity to get useful, robust estimates for females, males are kind of a freebie. There's a way of looking at that. So these simulations look at the different factors here, and looking at that image on the right, from top to bottom on the rows, the top row looks at variation in movement parameters. So basically think about that as home range size, going from small to large. The middle row looks at detection probability, again, going from low detection to pretty high detection rates. And then the bottom row looks at the number of sampling occasions. And so by running the simulations and iterating through different combinations of detection rates and so on, we were able to come up with some pretty good insights into study design. And these two are spatial in a sense. Another really tremendous advance with these methods is this idea of a clustering design. So instead of having a uniform grid of detectors across an entire region like we've done in the past, we're able to get the information the models need by looking at much smaller but kind of concentrated areas. So again, here the plus signs, the red plus signs indicate traps, hair traps in our case. But the question remained, what kind of intensity did we have to sample at for a desired precision of our estimate? Based on the information we had, which again was pretty solid, we determined that a 10 kilometer spacing between traps and about 50 kilometers between the center of each of those clusters was adequate spacing with 5 2-week sampling occasions. These produced, the simulations suggested unbiased and relatively precise estimates, in this case of around a CV of 12% or so. So definitely, useful estimates for the most part. I do want to note that the 2-week issue, the time is not important. It's 2 weeks is what it requires to obtain the detection rates that we used in the simulations. It's not the number itself, the time duration itself is not important. It's really, that's just a surrogate of sorts for detection rates. And in the end, this is more or less a design we ended up with, which for the sake of our budget and our field crews was highly advantageous. Speaking of field crews, our 2002 season was the big year, and this coincided with that mark resite surveys that I started off talking about with the aerial surveys. For this study, we used about 15 full-time field technicians, ran for about 2.5 months to allow us time for those 5 2-week sampling occasions. We had done pilot studies the previous 2 summers on this work and learned just a great deal about access issues. This is some fairly remote country, fairly inaccessible without relying on helicopters, which is very common in Alaska, but is not necessarily well suited for these kind of methods because then you're dependent on flying conditions. We stuck the boots on the ground almost entirely, and that allowed us to continue sampling regardless of field conditions, other than we did have some fires and then flooding and some other things that got in the way. But in the end, as you can see, we deployed about 605 hair traps, nearly half of which produced at least 1 hair sample, yielding over 2,000 hair samples total, over 600 of which were successfully genotyped. And that's a number we're pretty happy with. That's just grizzly bears. We also detected a lot of black bears, far more than grizzlies, in fact, but we're just talking grizzlies for now. So of those 601 samples, we identified 137 unique grizzly bears. You can see a pretty heavy female bias there, which for a bear population at carrying capacity or more or less stable dynamics, we do expect a slight female bias, maybe a 60-40 bias. What I want to note here is this is just raw data. These are not corrected for detection rates. Like say if we just detected females far more readily than males, that would not be reflected here. There's not that correction factor yet. Also very exciting with this, and what I mentioned one of the strengths of the seeker methods is we had 351 what I'll call spatial captures. So this may have distilled down to only, say, 200 detections in the sense of a traditional model, where it was just binary, did you or did you not detect an animal. So we have a lot more information to work with here, with those over 350 detections in space. And these were pretty evenly distributed over time. There was a pulse towards the end, which is also not uncommon as people learn their area better, just become more efficient with the methods, crews become more efficient working together, that kind of thing. But because of these models, which directly incorporate things like sampling efforts and allow other covariates, which I'll touch on more here in a second, we're able to help model that variation, account for it, so to speak, in a modeling sense, and not treat everything as being uniform across time and space. So we do have some very preliminary estimates, density estimates from these models. To be cautious, I'm not going to present them. I will say, however, that they are very consistent with our expectations for this population, given the management directive to reduce density. Again, that goes back 30 or so years. Further, the estimates from our hair trapping and spatial modeling were very consistent with the preliminary mark resite estimates from the collar data, although, as I've said, that 3,000 square kilometer study area is not necessarily representative of the entire region. So making direct comparisons requires some caveats, for sure. But we are very happy with preliminary estimates and are continuing to work on this with our partners and hope to get this published before too long. So next steps, it was important to think ahead. As I mentioned early on, one of the most important things we could do is to look at the variation in density in this population. So it's not just knowing that there are X number of animals in a given area at a given time, but trying to explain the variation in the population. As Jack mentioned in the beginning, I did my dissertation on a very large project in Montana. We had over 200 technicians in one season. It was $6 million. It was a bit of overkill. But what that allowed us to do was to model that variation in grizzly bear density. And this is just showing the Glacier Park area. That's the black outline there is Glacier Park, Montana. It's about 1 million acres, 4,100 square kilometers. So what I was able to do, because we had such a rich data set, was break the data into male and female, and then parse it into different seasons within a given summer. So I truncated this just to spring and early summer. But you can see this heat map shows the density of activity centers as a function of different covariates. These are from the best model. I ended up running over 1,400 models for this. It was, again, overkill, but just such a unique opportunity to have such a rich data set. Now, I don't want to confound this with what could be an actual density map, or certainly not a resource selection map. Although it is very, I say, enlightening to see how these patterns change over time. And having lived in this park for over a decade, I can say, and talking to many, many biologists, that these predictions, these estimates really do align with our understanding of bear ecology in this system. You can see there's a lot of overlap between males and females, but yet they do show some differences, and certainly changes over time as different resources become available. And if the later summer models were up here, you'd see that, for example, areas known to have high concentrations of grizzly bears, or sorry, of huckleberries, do produce higher density estimates for that season. In this case, I also modeled the effects of conspecific density. So I looked at female and male black bears as well, and found some very interesting results in terms of how those populations interact at a population level, not at an individual level. Another very powerful application of this is the potentially and very, very cautious extrapolation of results like this to areas that may not have been sampled. The study in Alaska example being the adjacent area of Wrangell St. Elias National Park, which was the southeast corner of our study area. Even though we did very little sampling in there, if we were able to find reliable ways to explain variation in bear density, say as a function of access to spawning salmon, that kind of thing, or caribou calving grounds, whatever it may be, if we have something that we can quantify to use to extrapolate to unsampled areas, that can be useful, again, if done cautiously. But that's something you really should not do if you only have a point estimate for a given area, to just extrapolate it without quantifying the relationships between quote unquote habitat quality and population density. Further, what this may allow is predicting how a population could respond to changes on the landscape. For example, if there were large wildfires, if bears were known to heavily utilize moderate burned areas, and we had large burns in the area, we might expect a positive response in the population, that kind of thing. Again, it has to be tied directly, though, to the sampling results. So one of the challenges, though, of course, is that developing and identifying these informative covariates is really not simple, especially given the heavy reliance on what's just available, using just the free global remotely sensed satellite imagery may or may not be reliable. It might work with your models. They might be supported in terms of model selection, but yeah, that's where you have to really ask yourself, are they really reflecting the process that you're interested in, or is it just either convenience or a surrogate for something else? Elevation is one of my favorite things to pick on. People love to use elevation in their models, but you got to ask, what is that really saying? I really doubt that bears are selecting for higher elevation in terms of like atmospheric pressure. It's the other things like temperature, maybe variability in the available food resources at a given time. It's not elevation itself, but people continue to use those things because they're available and it's commonly accepted. But you got to be honest about it. Ask yourself if those are really capturing the process you care about. And in terms of management, and it's even trickier if you're trying to find things that can be manipulated. If you were lucky enough to find a relationship between, say, density or population growth rates and wildfire, that's something that can be manipulated. Things like, well, elevation or access to salmon are really tough to manipulate. So it may be even more difficult for relevant management actions. So that wraps up the discussion of our ongoing research and kind of hopefully leads some questions from folks about how this relates to CWS in particular. And like Jack said, we're about to offer our introductory level class on capture-mark-recapture beginning here in a few weeks. And just to point out some of the background on our courses, these online courses are really developed to learn at your own pace, which is a great advantage. If we also have hybrid classes and in-person classes that are a bit more structured. But online classes are really geared towards students who maybe have a non-traditional schedule. These can be offered or provided in both instructor support and no instructor support, depending what your needs are. In my classes, at least, the things I really key into are asking the right question. Again, don't start with, I want to hang collars. I want to hear that we want to estimate survival rates of a given population. Identifying your objectives, defining your study population, which again can be very challenging. Animals rarely recognize geopolitical boundaries. There's no reason that they would stop just because there's a black line on a map. And then for me, hypothesis development is also critically important for much research. Not everything. If your question simply is looking at population growth rates, that may be just fine. But preferably working off hypotheses is really how we advance our understanding of these ecological processes. Another, I think, key, especially for the introductory level classes, is developing a solid foundation in these models and understanding relationships to potentially more familiar modeling frameworks like logistic regression, for example. But without having the foundation, I think it's really kind of dangerous, and I've certainly done it myself, to jump in at more of an expert level with some of these more fancy models, I'll say, like the seeker models, more demanding frameworks. Having a good foundation is really important, I think, before taking a leap into something more technical. Covariates, obviously a pretty close topic to home for me. We discussed what you can and cannot do with them, in part, at least discuss it. There's no absolutes. How to work with them, how to incorporate them into your models, some of the common problems with covariates. And then another, I think, strength of the class is that we discuss a lot of linking animal behavior and ecology, population status, life history traits, and so on to the sampling and analyses themselves. I think the population status is another question that doesn't get addressed nearly often enough, in that, say you're looking at snowshoe hares, we know they cycle dramatically, but if you were to do a standalone snapshot study, how do you know what point of the cycle you're in? And that can apply to anything. Is your population at carrying capacity? Is it experiencing a decline? Just these standalone studies, and there's some great examples out there that have shown the risk of these snapshot studies for bears and other species. Both classes, we'll discuss model selection theory. These are certainly part of the routines, the exercises we have as well, looking at assumptions and limits. It's not nearly as in-depth as, say, David Fletcher's course. He's truly the expert with these, but it is important to understand things like model selection and goodness of fit testing with specific context to market capture models. We also get into other applications and modeling frameworks such as integrated population models, using capture data. There's detection data to look at multi-state models, for example. That's actually how we develop projection models with the bear data, for example. And then particularly in the CMR2 class, we will do a much deeper dive into seeker models themselves, mark-recite models, which are a very similar, same family, and very, very powerful with some advantages over the more traditional market capture approaches. And then we're also going to have a co-instructor helping, who's much more a Bayesian expert than myself, to help bridge the gap, so to speak, between Bayesian and frequentist approaches, and see some of the relative pros and cons of each. Again, it's rarely black and white, which method is the best. Each method has its own advantages, whether it's just the technical aspect of leaping into the Bayesian world, or how reliable your data are, or how rich your data sets are, for example. Overall, CWS offers, I'm not even sure what the tally is now, Jack, I think over 50 courses, very specialized trainings, whether it's in person or hybrid immobilization classes. You see on the far right there, Dr. Daryl McKenzie leading an occupancy modeling class. Again, these are either in person, online, or hybrid, with the goal of providing everyone accessible opportunities to develop the skills, knowledge, and abilities, whether it's trying to advance your career, get into a graduate program, just personal understanding. That some of this is more interesting than maybe promotional material. I'd like to think some of the bear material it's very exciting stuff. And we also provide group trainings if your organization has a larger need than just a few individuals. I know we'll go into the structure of this. All this information is available on the website, but there are just a plethora of options for students and how they access materials, the different levels, whether you're trying to or desire having academic credits, continuing education credits, professional certifications. Life Society and Ecological Society of America are both partners. So there's lots of options for students. It's a very, very flexible format. With that, again, I encourage people to check out the classes online, and I'm going to hand it back to Jack. If anyone has questions about courses, bear research, Alaska, maybe, I'm happy to give it a shot.
Excellent. So I am just right now, I'm just throwing a few links for folks just to make it easy on you for these courses so that you can get there, and maybe while Jeff's answering some questions, you can have a look. And if you have any questions specifically about the courses, then you can go ahead and ask Jeff himself. But with that said, if we have any questions specific to the talk, the methods that Jeff talked about, if you have anything in specific that you'd like to say, we don't have a huge group here. So I think I'll just leave it to you to unmute and go ahead and ask Jeff. There he is. Anybody have a question related to the talk?
I think Forrest is speaking, but I can't hear anything.
Can you hear
there
me?
We go.
There
we are, Forrest.
Awesome. Just a hair abstract of a question. You in your course, the CMR one, and then Dr. Daryl McKenzie, I think he's the one doing the occupancy course. You guys spend a whole module on the hypothesis before field work, study design in particular before going out there, like you said. And I've worked before going to grad school as a technician for a ton of different federal and state agencies doing stuff. Where I've noticed maybe substandard scientific monitoring or processes that might be done by like Bureau of Land Management or something like that. And that's what I mean abstract. Have you experienced that kind of stuff? And do you know what's a good way to reorientate a field office to start using more of these kind of rigorous standards, do you think?
Yeah, it's a fantastic question and a really difficult one. And I'll try and keep my personal opinions to a minimum. But having worked with a state management, and I've been fortunate to work everything from academia, nonprofits, consulting, state agencies, federal agencies. And I think the challenge really is getting information into the hands of decision makers so that they can understand it. Many of these folks are not trained biologists, they're not trained scientists, they don't understand things like, and I'm not being insulting, it's just something that folks aren't trained in, generally speaking, how to interpret uncertainty in estimates, for example, or assumptions and models, or the complexity of all these interacting processes. So I think making a claim that information will be more relevant to decision makers is the way to go, because it doesn't matter how great your science is if it doesn't address the questions that they care about, things like predation rates. They're likely to not care so much. Another one I think is efficiency. I've tried to make the argument here, or tried to, that if you're not doing the right thing, you're doing the wrong thing. And so if you have limited resources to invest in research and management, it's really in everyone's best interest to identify ahead of time the objectives that actually matter. The grizzly bear study I referenced for my dissertation work, for example, I think that's maybe the best example I can think of. I was, one of many people involved with that. Just very lucky to be towards the top of the heap. We had 12 cooperating agencies involved with that. 5 national forests, national park, two native tribes. They all came together as a group and decided what the highest priority was for information about that population. And everyone was on the same page. We all had the same marching orders, and it worked fantastic. I couldn't argue a better approach than having people come together ahead of time and agree to what they need. I'm not saying they were right, but that was, at least had consensus. So I think trying to build consensus among the decision makers and stakeholders. Otherwise, we're just talking to ourselves some of the times. Yeah.
Thank you.
Jeff, there's a question from Lydia.
Okay.
She gave you some praise on the talk, and then she's got some interest and probably some concerns about genotyping error. She wanted to know if you use SNPs. And, how do you potentially identify those errors, those genotyping errors? What kind of strategies might you use to get at that sort of thing? And there was an example here, like if you had two different individuals in the same sample and the DNA was combined, for instance.
Yeah. Yeah, thanks, Liddy. It's a great question. And I will say, 10, 15 years ago, that was a huge concern, where things like degradation rates, Scott Mills' shadow effect, for example, if you don't have enough power in your genotypes to differentiate individuals, they end up looking the same. And that actually is a double whammy, just to throw that out there. If you have two animals that look the same genetically, not only are you decreasing your number of animals in your sample, but you're decreasing their detection probability. Sorry, you're increasing their detection probability. So it really is a double whammy when that happens. In our case, no, we're still using microsatellites. We worked with Wildlife Genetics International to make sure our suite of markers had more than enough power without getting into that realm of having so many markers. You increase the risk of generating spurious genotypes. And that's David Petkow's bread and butter in large part. His paper in 2003 talks a lot about that. Trying to optimize your marker selection. There was a lot of debate in the literature back then, but I highly recommend Dave Petkow's paper on that. And specifically, again, with that large study in Montana, we did, I submitted, I want to say it was 117 mixed samples from either full siblings or parent offspring pairs. The lab knew we were submitting them, but they had no idea what the genotypes were, what samples were combined. They had no idea. And even though it was a relatively small sample size, we would argue small sample, that project collected 32,000 hair samples. So that 120 or so samples was a pretty small proportion, but we had zero errors. There were no spurious genotypes detected or generated. Either the sample failed outright, we were delivered from the lab one or the other of the correct genotypes that were used in the mixing, or the lab said it was a mixed sample. There were no questions about it. So SNPs, I know a lot of people are moving to SNPs for some questions. Like I helped with a river otter SNPs for spraints because it's such a hostile environment for DNA. But for hair samples, microsats are still working pretty well. With that, it's a function and part of your sampling interval. I have a buddy and worked in New Mexico, had just atrocious genotyping success rates because of the UV load. It just baked the samples essentially and had very, very poor success. So you have to, all those are a lot of things to factor into it. SNPs may work better for him as well. But so far we've had, I think, very little concerns over things like mixing, just due to the quality of the lab we work with. And if that didn't address it, please let me know.
Any other questions out there?
And we worked with Lisette Waits in the beginning years of the work in Montana, and then moved up to WGI when Petkow established that lab. Yep. And so the work I mentioned, or the testing of the lab, that's in a paper, Kendall et al. 2010. I want to say, I should know. That was the primary write-up for that big project in Montana, where we discussed the mixing in the lab results.
Feel free to follow up with Jeff after the talk if you want actual citations. I'm sure he'd have no problem sending those off to you. He's got them all right there on his desktop.
Yeah, happy to, for sure. Always love talking about bears and genetics. Yeah.
That's right. That's right. Anybody else have any questions? All right. Well, Jeff, thank you so much for being here, folks. Please feel free to reach out to myself and Jeff about CWS and this talk and these methods. We'd be happy to answer any questions that you have. And thank you for coming. I know it's the end of the day for many of you. Thank you for sticking it out. And Jeff, thanks again for a great talk. And we look forward to seeing you in the classroom, hopefully.
Yeah, thanks again to everyone for tuning in.
All right, everybody. Take care.